Hi learners, welcome to Commissionerate of College Education, Vijayawada. I am Dr. K. Daniel, HOD of Zoology, JKC College, Guntur. I have been explaining the cloning vectors. Previously, two video lessons have been uploaded in my YouTube channel and also in the LMS CC office. And in this lesson, that is in this video lesson 3, most probably I would like to explain the last part of the cloning vectors. So it is the heading is cloning vectors 3 like that. So let us see quickly the contents of this video lesson. The first one, it is about the cosmid vectors, and the second one is the back vectors, and the third one is the ag cloning vectors. First, cosmid vectors. So these are the headings of these cosmid vectors. First one is introduction about the cosmid vectors. Second one is the structure and characteristics features of cosmid vectors. Third one, examples for cosmid vectors. And fourth one is the steps to cloning of foreign DNA into these cosmid vectors. And fifth one is advantages of the cosmid vectors. And the last one is the disadvantages. Okay. So let us see briefly one by one. The first one, introduction about the cosmid vectors. What are the cosmids? Cosmids are nothing but cos sites of phase vectors and plasmid vectors. So I will explain later on. First, who discovered these cosmid vectors? These were first discovered by Collins and Horn in 1978. These are the medium-sized vectors. They were developed by combining both the features of bacteriophage vectors and plasmid vectors. So here cosmids is equal to cos means cos sites of the phase vectors, mid means plasmids are a part of plasmid vector. Then the structure and characteristic feature of cosmid vectors. These cosmid vectors are the hybrid vectors derived from the plasmids with ORI, origin of replicate sequence, and a fragment of lambda phase DNA with its cos sites. Then what are the cos sites? Cos sites are the sequence yielding cohesive ends which allow the foreign DNA to be packaged into lambda phase heads. That means to package the foreign DNA into a phase vector or into a bacteriophage head, the cos sites are very important. These are nothing but the base pairs or the DNA sequence which have been taken from the bacteriophage vectors or bacteriophage virus. A cos site may have about 12 base pairs. It may have the length of 12 base pairs. Then, what is the typical structure of a cosmid vector? What are the parts present in it? So, a typical cosmid vector contains the following parts. Number 1, ORI, origin of replication. Number 2, MCS, multiple cloning sites. They are also called polylinkers for site for the restriction agents, and at least a selectable marker that may be AMPR, ampicillin resistance marker. And fourth one is the causites. The causites are derived from the lambda bacteriophage virus. Okay, you can see over here the picture. So this is the cosmid. Both it is nothing but the combination of both the causites of the phage vector or phage virus and plasmid, a part of plasmid vectors. Okay, here you can see the ORI, the causites. Causites are nothing but the cohesive ends which are taken from the bacteriophage vector or bacteriophage virus. And this is nothing but polylinker which is called MCS, multiple cloning site. This is for uh, ampicillin resistance sites, antibiotic resistance site. So in addition to this typical structure, the cosmid vectors have the following properties or the characters. Each cosmid vector or a cosmid is circular with a double-stranded DNA. It is a circular with a double-stranded DNA, but it has two complementary single-stranded regions at both ends of the plasmid DNA. The two cos ends form a duplex by base pairing. So causites are the only requirement for packaging into the phase particle. So they are desperately needed. What are needed? Causites are needed for the packaging of foreign DNA into the phase head. Without causites, we cannot send the foreign DNA into the host cell. That you have to remember. So cosmid DNA does not integrate into host chromosomal DNA and replicate independently. This is another character of cosmids. That means they won't integrate with the host chromosomal DNA. They can replicate or multiply separately in the host cell. So one type of character which is taken from the bacteriophage. You remember that a bacteriophage has two life cycles, lytic phase and lysozymic phase. So it is a type of lytic phase where it can replicate inside the host cell, but it won't integrate with the host chromosomal DNA. Host is nothing but E. coli, Escherichia coli. And another character is it has the length of about 6 kb, kilobytes, and can be inserted up to 40 to 45 kb of RNDM. So when compared to the previous ones, it is more larger. It is more larger than plasmid or bacteriophage vector, which you have discovered, which we have discussed in the previous lessons. Okay, so it is more advantage because it can insert up to 40 to 45 kilobase pairs of RNDNA into its own DNA. And a packaged cosmid has the character of both infecting the hosts like lambda phase and can multiply like plasmid. This is a very important character. So if you want to construct a cosmid vector means it is having both the characters of bacteriophage and also the plasmid. What is the character of this lambda phase or bacteriophage? It can infect the host cell or it can infect the E. coli host cell. At the same time, it can multiply just like a plasmid vector inside the host cell. So there is a dual function of this cosmid vector. Hence, they are more advantageous than the plasmids or bacteriophages. And another important property is they can grow both in bacteria and also in other eukaryotic cells or animal cells. Next, the third one. So I have given two examples regarding the cosmid vectors. The first one is the cosmid cosmid PLFR5. So this is This is the cos sites. Cos sites are nothing but cohesive ends of the bacteriophage or lambda bacteriophage. Okay. So this is the MCS, multiple cloning site, origin of replication. And of course here in this example, instead of ampicillin, there is another antibiotic resistance site, that is tetracycline resistance site. So this example, that is cosmid PLFR5, is the commonly used cloning vector suitable for cloning large DNA fragments up to 40 kilobase pairs. But of course, it is having the size of 6 kb bytes in size. It is constructed from E. coli plasmid PBR322. P means plasmid. And two cos ends of lambda DNA. So both plasmids and cos sites of the lambda phase. So this plasmid derived portion contributes an origin of replication and tetracycle resistance gene. Okay. A foreign DNA of up to 45 kilo base pair can be inserted into the MCS multiple cloning sites of this particular cosmid vector. The second example is cosmid PJB8, but of course it is also derived from both the plasmid and cos sites of the lambda phase, but it is having origin of replication. Instead of tetracycline, it is having ampicillin resistance sites. Okay, so it can insert up to the foreign DNA up to 45 kilo base pairs at different resistance sites like bomb H1, E1, E1, E3, like that. So these are the two examples for plasmid vectors, which are frequently used in as tools as cloning vectors. Then, what are the steps for the cloning of foreign DNA into cosmids? So these are the 5 important steps. First, we have to do the first step, that is the ligation of the foreign DNA between two cos sites. That means we have to take two cohesive ends of the particular vector. Then we make a concatenaric DNA. Concatenaric DNA is nothing but the long chain of the DNA, which is repeated or multiple copies of DNA is present. So these concatenaric DNA, it is possible to make the concatenaric DNA due to the presence of two cos sites and the ligation of these two cos sites with the foreign DNA. And third step is in vitro packaging to introduce the DNA into phase head to form the mature phase particles. It is nothing but packaging of the foreign DNA into lambda phase head so that it becomes a phase particle. Then this phase particle should be introduced into the host cell by a process called transduction. So after their entry into host cell, the cosmids are maintained as plasmid. Okay, these are the 5 steps you can see here in the diagrammatic representation. This is the cosmid. You can separate the cos sites from the multiple cloning sites, that is cleavage at polylinker site or multiple cloning site. This is the target DNA which we want to multiply, and these are the cleaved cosmid cos sites. So between the two cos sites, a foreign DNA is ligated. Okay. Now this ligated foreign DNA, along with packaging into the vector, it will be introduced into the host cell for the multiplication. So those are the steps for cloning of a foreign DNA into a plasmid or a cosmid vector. Now, the fifth point in cosmid vectors, advantages. There are so many advantages. But of course, the first one is these are comparatively or relatively larger DNA fragments when compared to plasmids, as I told you previously. So they can insert up to 45 kilo base pairs foreign DNA into it. This is as it is pick up or it picks up large DNA fragment. They are used to establish gene libraries. Gene libraries. And another important advantage or use is gene cloning through plasmids helps in the study of nonsense sequences in the genome of organisms. There are, as you know, there are some coding sense and non-coding or nonsense sequences in a particular genome of a particular individual. So these plasmid vectors can be used to study such type of nonsense sequences. And another advantage is some plasmids are constructed by joining a linearized plasmid DNA with DNA fragments of P1 bacteriophage. This is another bacteriophage or bacteriophage virus which have the causates. So they are used by joining the linearized plasmid DNA with the DNA fragments of that particular vector. Okay, so like that there are so many advantages. What are the advantages? They are more larger than the DNA fragments, and they are used for establishing the gene libraries. Not only for that, they are also useful to study the nonsense sequences and bacteriophage vectors also. A special type of bacteriophage, that is P1 bacteriophage, which can join the linearized plasmid DNA with that particular DNA fragment of the vector. Now, the last one in this plasmid vectors is the disadvantages. There are definitely disadvantages, because if there are advantages means definitely they have some disadvantages. The first one is the packaging enzyme fails to pack recombinant plasmids into the phase head if any one of the two causates is missing. That means for packaging of a foreign DNA into the phage head of a particular virus, the necessity of having two causates is essential. So if the causates are missing, any one of the any one of the two causates is missing, definitely it fails to pack the recombinant DNA into the plasmid vector. Sometimes more than one recombinant plasmid join together to form large DNA. It is another disadvantage, and they are slower in replication, and they are unstable. Sometimes they are unstable inside the host cell. This is another disadvantage of plasmid vectors. So this is about the plasmid vectors. Okay. Next, the second one is back vectors. So what is back vector? Back. There are two important vectors, back vectors and act vectors. So before we go into details of these back vectors, first let us learn about artificial chromosomes. Then we'll go about back vectors and the structural components, applications, so on and so forth. Then, what are the artificial chromosome vectors? What are the artificial chromosome vectors? These are DNA molecules assembled in vitro from defined constituents which guarantee stable maintenance of large DNA fragments with the properties of natural chromosomes. With the properties of natural chromosomes. So artificial chromosomes are nothing but DNA molecules assembled in vitro from defined constituents, which guarantee stable maintenance of large DNA fragments with the properties of natural chromosomes. So they are useful for genome sequencing programs, for functional characterization of entire genomic regions, and also for the transduction of large DNA fragments into human and non-human mammalian cells. So you remember, the artificial chromosomes are nothing but DNA molecules assembled artificially from a special type of DNA sequences. It is nothing but DNA sequences. Let us see, there are so many artificial chromosome vectors have been created, but here just I would like to explain only two, that is the wax and X. X and X. This is the first one, bacterial artificial chromosome. It is the elaboration of wax. Wax means bacterial artificial chromosomes. What are the bags? A bacterial artificial chromosome is a DNA construct based on the functional fertility plasmid used for transforming and cloning in bacteria, usually E. coli. So in E. coli, there are two types of plasmids. What are the plasmids? These are the extra chromosomal DNA present inside the E. coli cell or bacterial cell. So F for fertility or fertility for E. coli plus. That means if the F or fertility plasmid are used for transforming a particular or cloning bacteria, from that particular plasmid we can construct this type of artificial chromosome. That means a plasmid with F plus means male. F plus it indicates maleness, whereas F minus means femaleness. So the fertility plasmid is used for construction of a bacterial clone, that we have to remember. So these bacterial artificial chromosomes are capable of carrying approximately up to 300 kilo base pairs of inserted DNA sequence. Here you can see this is the plasmid. So these are more larger than the cosmos. Rather, you can say they are more larger than the cosmos. Then what are the structural components of bags? So generally it is the artificial constructed vector, as you know. There are different parts which are taken from different vectors or different DNA sequences. Let us see one by one. First you can see here is a ORI, origin of replication, origin of replication. And there is another part which is called repi. Just wait. It is a plasmid replication and regulation of copy number. And there is another part, par A and par B. These are two parts, they are meant for the division of a particular DNA or plasmid DNA into daughter cells. These par A and par B. And there is another part which is called selectable marker. Selectable marker. So these selectable markers, they are nothing but antibiotic resistance sites. Some bats may also have lag gene in addition to selectable markers. So these all these parts they have been taken from the plasmids, taken from the plasmid. And there is another site which is called T7 and SP6 phase promoter for transcription of inserted genes. For transcription of inserted genes. So these are the common gene components of a bag. What is the first one? ORI. ORI is a part. And another one is REPE. Third one is PAR-A and PAR-B. And fourth one is the selectable marker. Selectable marker. And the fifth one is nothing but unique restriction site. They are nothing but multiple cloning sites, rather multiple cloning sites which are having lag gene and T7 and SP6 sites and also cause sites and also cause N sites. In this region, cause N sites are also present. This region is nothing but unique restriction site which is called MCS, which is called MCS. These are the structural components of a bacterial artificial chromosome. Now we will see quickly about the applications of BACs. So the first and foremost significance or application of BAC is they are used as models of disease. They are models of disease. Say for example, these BACs have been used to study neurological diseases like Alzheimer's disease, Down syndrome, so on and so forth. And another important application to use these BACs are infectious diseases. Or they are used to detect the infection or to study the infectious diseases. Say for example, to study the genome of several large DNA viruses, RNA viruses, which infect the human beings as well as animals. So these are the two or other important applications of BACs. And as I already told, there are so many advantages. One of the important advantages, these are larger. They can insert larger DNA molecules into its these back vectors. Okay. Now the last one, the third cloning vectors, which I am going to explain briefly, is YAC vectors. YAC vectors. These are the headings which you have to remember. First, we will see the introduction about YACs, structural components, construction of YACs, applications of YACs, limitations of YACs, that is disadvantages, and in the last we will see the differences between BACs and YACs. Okay, so let us see one by one. So just like BAC vectors, these are YAC vectors. That means yeast artificial chromosomes. YACS, yeast artificial chromosomes. These are genetically engineered chromosomes derived from the DNA of the yeast. You know about yeast, no? So here, the type of yeast which to be used is Saccharomyces cerevisiae. This is the particular species which are used in the construction of these acts. Okay, yeast artificial chromosomes allow the cloning within yeast cells and of fragments of foreign genomic DNA that can approach at least 500 kilo base pairs in size. 500 kilo base pairs in size. That is more larger than the back vectors. Then how they are constructed? How they are constructed? So the construction, it is mainly built using an initial circular DNA plasmid. That means for the construction of AG, in addition to the yeast DNA molecule, they also use these DNA, circular DNA of plasmid. So this circular DNA can be cut into linear DNA molecule using restriction enzyme. Then DNA ligase is another enzyme to use it to ligate the DNA sequence or gene of interest into the linearized DNA, forming a single large circular piece of DNA. So generally, YAC vectors are linear, but they can be made circular by ligation of foreign DNA into it. Okay. Before that, you will see, or I will explain what are the structural components of these X. Structural components of the X. As I told, there are two types of DNA sequence can be added to form an act or yeast artificial chromosome. What is the first one? First one is regarding the yeast chromosome, that is Saccharomyces cerevisiae, and the second one is the DNA sequence of the plasmids or bacterial plasmid. Okay. So first, let us see what are the structural components of this YAC by using or by taking the yeast chromosome. Okay, the first part of this act is the yeast centromere. This one, yeast centromere. It is designated at CEN4 like that, CEN4, and it is allowing. It is useful for regulation of the segregation during mitosis. So the centromere is useful to regulate the segregation of the chromosomes. And the second part of this YAC vector is yeast ARS. ARS1. You can see here, this is the ARS1. ARS1. So this part of DNA is useful for replicating of the vector, that is yeast cell. To replicate the yeast cell, this part of DNA is essential. And third part is the yeast TEL. Here you can see TEL means telomere. Telomere is nothing but a part of the chromosome. No? It is the sequence of the chromosome. So it will convert the circular plasmid into linear plasmid. This TEL or telomere is the part of the yeast cell. It can convert the circular plasmid into a linear plasmid. And fourth one is the gene for ACT selection in yeast. Fourth one is the gene for selection in yeast. That is nothing but selectable markers. Selectable marker over here. Selectable markers. And these are the four important parts or the components of ACT which is derived from the yeast cell. Which are derived from the yeast cell. And there are other 2 or 3 parts derived from the bacterial plasmid. Bacterial plasmid. So they are number 1, ORI, ORI, origin of replication, it is common, ORI. Here you can see ORI. This is derived from the bacterial plasmid. And of course, selectable markers, bacterial selectable markers over here. And the third one is MCS, multiple cloning sites. See for you like this, no? This is the multiple cloning. These three parts are derived from the bacterial plasma. So, YAC vector is nothing but the combination of both yeast chromosome as well as the plasmid, as well as the bacterial plasmid. Now, this is the, of course, these are nothing but the steps involved during the cloning of a particular foreign DNA into the YAC vector. So these are the green colored, these DNA sequences are nothing but the gene of interest, which we want or which we can replicate them inside the vector. And these are the parts of the YAC vector. Just I explained about this. And we can cleave them. The circular DNA can be cleaved into linearized DNA by using restriction enzymes. So because of cleavage, there is a linearized YAC components arise. In between these two ends or in between these two parts of the YAC vector, we can insert a foreign DNA. This is the foreign DNA. So it will be integrated over here and it will be transformed. For the transformation, it will be inserted into the host cell. Okay, just like what the cloning methods we are used are being used in the animal cloning, what you call cloning methods. Okay. Then, what are the applications? Yeast expression vectors such as YACS, Y1PS, yeast, YIPS, yeast integrating plasmids, and yeast episomal plasmids have an advantage over bacterial artificial chromosome in that they can be used to express eukaryotic proteins that require post-translational modifications. So, yeast chromosome or artificial chromosome vectors are used as expression vector. That means they can express in the process of protein synthesis even after the translation. That means even after the formation of the protein, we can use these tags as expression vectors. So vectors are of two types now. This is one type, that is expression vector. There are some other vectors which they won't express. They won't express. Only they will be useful for replication or multiplication of the foreign DNA. Okay. And these YAC vectors also useful for physical mapping and also chromosomal walking. This is another one type of mapping of the chromosomes which are being used in the Human Genome Project or HGP. Okay, so they are useful for chromosomal walking and also chromosomal mapping. And there are certain limitations regarding the YACs, of course. They are, even though they are very large, they are very fragile and prone to breakage, leading to problem of rearrangement. There is a problem of rearrangement in these vectors. So even though they are very large, but they are very fragile and they are prone to break. And there is high rate of loss of the entire yak during mitotic growth. This is another disadvantage. And difficult to separate yak from the other host chromosome because of their similar size. Like that, they are having advantages and also disadvantages. And of course, this is the last slide. It is the comparison between backs and acts. So as far as the configuration is concerned, backs are circular, whereas acts are linear. And as far as the host is concerned, for backs, bacteria is the host, whereas for acts, yeast is the host cell. And the copy number it is somewhat low, but 1 to 2 in backs. And the cloning capacity is unlimited in acts, whereas it is very low in backs. And they have a chimerism, that means changing into changes will be happen. Changes will be happen because they are sometimes they are unstable. And insert stability is stable in backs and unstable in acts. So like that, the cloning vectors are nothing but DNA molecules which carries the foreign DNA inside it. So they are very useful and used as tools in the animal biotechnology. Okay, learners. So with this, I complete the cloning vectors. So there are 3 series: cloning vector 1, cloning vector 2, cloning vectors 3. So these 3 video lessons are uploaded in my YouTube channel. So you can watch and you can subscribe and you can like it. Okay. Thank you very much. Thank you one and all.